Hello, everyone. Welcome to the Demyelinating CMT types breakout session. I'm absolutely thrilled that you have all stayed in the room. Thank you ever so much. If you'd like to take your seats. We have three really great speakers. I'm so thrilled that you could all give up some time on their Sunday to talk to you here. We're going to be hearing first from John Svaren, who's chair of the CMTA Scientific Advisory Board. A wonderful, wonderful person to work with. And we also have Vera Friedman, who you heard from yesterday, who's at the CMTA Center of Excellence here in Denver. And we also have Rachel from Armatus Bio. So we'll have a biotech perspective on what's happening in type 1 research. So now that I've got you all settled, I'm going to hand straight over to John Svaren, who's chairing this session. Thank you, John.
Good morning. It's really a pleasure to be here. My name is John Svaren. I'm at the University of Wisconsin in Madison. I have a lab that studies Schwann cell development, but we also study CMT neuropathy, and we work with a number of different scientists on different aspects of the disease. A couple things I want to say. My most important role here is to really introduce two other great speakers. But I do want to give you sort of a flavor of the scientific enterprise as it pertains to CMT. One thing you may not know, there's literally hundreds of scientists around the world who are working on this disease. We had a meeting in Montreal back in June and really saw the latest and greatest that's in this entire world of research. And we as the CMTA have tried to recruit the best and brightest scientists to be part of our advisory boards and also to undertake different projects as well. At the end of the day, I was inspired by seeing Dr. Friedman's team yesterday, and I think it really reinforces the idea that we need to rely on a team approach for CMT. So we work with not only within scientists within our network and neurologists, but we really aim to partner with companies and other entities that can really bring us new cures for CMT. So forgive me if I start off with some basics here, but I did want to remind you that you're in this session because presumably you might have some interest in the demyelinating form of CMT. And just to sort of make that clear, we think of peripheral nerves as sort of a wire, which Sue mentioned earlier. And on the right, you see that bundle of wires. And you can think of Schwann cells that make myelin as the insulation around those wires. And demyelinating CMT essentially affects that insulation. Now, Schwann cells are more important than that insulation. They actually make nerves transmit faster. So without Schwann cells and myelin, nerve transmission would be very slow. And sometimes I like to think of this as Schwann cells making the transmission of nerve impulses, making it from 3G to 4G. And some of you have 5G phones, which are even faster. And so Schwann cells really play a very important role with that. In the demyelinating form of CMT, we have deficits in those Schwann cells that we're really aiming to try to fix. So these are classified as CMT1 or sometimes CMT4, and this is distinct from axonal forms of CMT, where the principal cell that's affected is really motor neurons. So these are the general, most common types of demyelinating CMT, and CMT1A is caused by an excess level of a gene known as PMP22. This makes an important protein that's a constituent of myelin. And we know that the dose of this protein is incredibly important because if you have too little CMT1A as a result, you can have, if you have too much CMT1A as a result, but you can also have too little of the protein, and that causes another disease known as HNPP. The other types of CMT are caused by mutations in other important proteins that are part of myelin. So mutations in the GJB1 gene cause CMT1X. Mutations in myelin protein 0 cause CMT1B. And these are really some of the very most common forms of CMT. Okay, let me try this again. Okay. And then another relatively common form is CMT4C, which is caused by mutations in the SH3-TC2 gene. So these are among the most common. There are some other forms of demyelinating CMT, but these are really, the bulk of individuals within our community that have this demyelinating form of CMT. So as Sue had mentioned, we're really trying to pursue a multi-pronged strategy for demyelinating CMT. And a number of those facets of our plan of attack involves really trying to take advantage of newer genetic therapies like ASO and gene therapy. We are also looking at drug development, really trying to identify compounds or drugs that would have promise for treating CMT. Another important element is really enabling clinical trials. One of the things that I've learned over time is that even if we had the perfect cure for CMT, it wouldn't really matter unless we have a way of doing an efficient clinical trial. And Dr. Friedman will talk more about in the next session. And then another really important part is really developing company partnerships, which really help us to tap into resources that are required to bring new cures to the clinic. So I'll just say a little bit about some of these genetic therapies of CMT. And the pathway for this was really laid out for us by work in a disease called spinal muscular atrophy. SMA is a disease that affects the same motor neurons that are affected in CMT. However, in this case, oftentimes this can be a very devastating disease affecting very young children, and oftentimes when it was untreated, leading to premature death. And work from a lot of scientists resulted in a couple new therapies for SMA, initially an AAV gene replacement therapy, and then an ASO therapy. And these treatments were spectacularly successful and are still being used in the clinic and then being extended to additional individuals with different forms of SMA. And this really was an inspiration for us to really try to pursue the same path for CMT. And there have already been some clinical trials for this kind of therapy in a type of CMT known as GAN. But one of the things that's very exciting is really some really great news that will be brought to us by Rachel Saltzman, who will be giving the third talk in this session, really talking about the development of a new gene therapy for CMT1A. We also have funded efforts to attack CMT1X and CMT4C in a similar way, and we'll cover this in a little more detail in the Gene Therapy 101 session that's coming up as well. So again, there is a spectrum of different types of gene therapy, ranging from genome editing or CRISPR-Cas9. Many people have read articles about that. We'll talk about that more in this extra session. There's other types of therapies called ASOs, stands for antisense oligonucleotides, and RNAi, and this is another important area as well. And then using this sort of AAV gene therapy approach, this is going to be the subject of the third talk, and this is really also a very exciting development. One of the things when we talk about gene therapy, there are three important things to know about gene therapy. That is delivery. In order to get this therapy in the right place, in the right cells, in our case, Schwann cells, if we're looking at demyelinating CMT, it's not a simple problem. And so developing technology to enhance this delivery to Schwann cells is really crucial. And this is really an active area of work that we're supporting as well. And there are many efforts ongoing for that. So I did want to tell you more just about also candidate drug testing that we're supporting from the CMTA. Certainly a big part of that is drug testing with pharma partners. We have this preclinical testing service, so companies that have molecules that are of interest, they can come to us and we can put it in a mouse that has a form of CMT and see if that improves that. And that's really been a great resource. That's really the basis of a number of different partnerships. We have scientists that are working on drugs to promote clearance of proteins that are causing the CMT. There's an effort sponsored by the Pugliese Laboratory that showed some positive results in CMT 1B and 1E. My own lab has a research project to try to reduce the level of PMP22 using a candidate compound. And so that the project is ongoing as well. We have another effort where we're testing drugs to stimulate removal or degradation of PMP22 levels, again, to bring that level down into the normal range. And then we also have another scientist of our board, Maurizio D'Antonio in Milan, who has an active effort in CMT1A and 1B to test drugs in a special pathway known as unfolded protein response pathway. So I don't want to imply that we really are relying on genetic therapies exclusively. We really have a spectrum of efforts across different modes of therapy in order to try to find something that's going to work and something that we can bring to clinical trials. So another really large part of this effort is we've realized that like if you are part of a business and you're starting up that business, you need some metrics to know whether your business strategy is actually working. It's the same thing in clinical trials. We need metrics to understand the status of the neuropathy and to know during the course of the clinical trial, are we actually improving things? And that's one issue and challenge that we've had in CMT is that CMT is typically classified as a slowly progressive disease. And what that means, though, is that sometimes the response to a therapy may also be slow as well. So we've been really working on developing more sensitive ways of assessing a neuropathy so that we can see early on in the clinical trial whether or not that trial is going to be effective. And this is incredibly important because companies that want to sponsor clinical trials, they need these metrics in order to invest in doing that. And this is really important part of our efforts to partner with companies is making sure that they can plan an efficient clinical trial. And Dr. Friedman will talk a little bit more about that in the next talk as well. So I did want to say that, repairing CMT or curing CMT is really a multi-step process. In our case, we need to sort of restore the myelin in Schwann cells. This is going to improve nerve function and nerve preservation. And then hopefully patients will notice. And that means that you'll have, ways of assessing whether activity or different activities are working better. And we're looking at trying to identify a lot of more sensitive measures along this pathway so that we can track this progression earlier and earlier. And so we're sponsoring efforts to not only do this in CMT1A, but we also have major projects where we're looking at CMT1B and CMT1X and among other types of CMT in order to sort of build these other metrics in to our clinical trial planning strategies as well. So with that, I just want to reiterate that we're really trying to integrate these efforts across the board, and I already mentioned these 4 different ways that we're doing that. And I also want to harken back to the fact that many times we get questions from people about what they're doing for their type of CMT. And that's a very legitimate question. But I do want to reinforce this is that we are really on the same team. And many of the activities that we're doing really have impact across many types of CMT. And there's many types of convergence in this way. One of this is development of gene therapies for CMT. If we develop that technology for one type of CMT, this is going to enable technology to come to the other types of CMT. Doing genetic diagnostics has really been critical. It's really been the basis of that success story that we heard for the keynote this morning. And we're supporting those efforts to really try to identify those subtypes, those genetic classifications, because that's very important. The biomarker effort, again, it's going to be important across multiple types of CMT. So this is not really a unique thing. And then also axon degeneration. So when we talk about CMT, what really leads to the progression of that is not so much that the myelin is bad, but over time, the axons will sort of deteriorate a little bit over time, and that really what drives the progression of CMT. So we're really looking for cures that may address that. because that really has the potential to work across multiple subtypes of CMT. On the basis of the work that we've done so far, I just want to say that there's really a high level of effort across the scientific community in CMT. And I want to thank you for coming today. You are really an inspiration for us. So your questions and your engagement and your presence here is really an amazing, inspiration for all of us as we work at many levels to sort of attack this disease. So we're going to have a Q&A session later, but I want to reintroduce Dr. Friedman, who you met yesterday, and she's going to talk more about clinical trial planning for CMT.
Hi, do you hear me okay? All right, hello again. So I want to start and just thank the CMTA again. I think I'm just going to keep doing that with each of my talks. I think it's an invaluable infrastructure and support system for CMT research, and we wouldn't be where we are without it. Okay. Here. All right. Okay. So as we just heard, and as I think many of you know, we're sort of standing at a very important pivotal moment for CMT research and for patients with CMT. And so that's particularly been true in the demyelinating forms because CMT1A is the most common form of CMT, and there's been so much research momentum in this area. But if you look at the picture over on the left of the myelin, you can see that several of the most common genes in CMT are myelin-based genes, right? So they're hugely important. And then on the right, what I want you to appreciate is just like the schematic of all of the treatment that has been arising in the preclinical space and headed towards patients. And this includes everything we just heard about: gene therapy, RNA interfering therapy, small molecule therapy. And it's really an explosion of sort of potential treatment interventions. This is an incredibly exciting moment. As a clinician, I feel very sort of humbled to be living this. And as a patient, I think there's a lot of promise for the first time. And that said, I think it's unearthing a lot of the challenges that we're going to have to tackle in thinking about how to actually bring these treatments to people and developing clinical trials. And I think it's critical that we start thinking about it now, as early as possible and as honestly as possible, to think about what we can do to address some of these challenges. So, we can talk a lot about the animal model work, but what we all know is that humans are not mice. And that's true in many different ways. And so when we're really thinking about whether a treatment is going to work, we have to think about a person. So I'm going to tell you the story of a recent clinical trial in CMT1A. There may be people in the room who've been part of this trial. I think it's a very important trial to talk about. And this was the study of PXT3003, which is a 3-drug combination. So they looked at baclofen, naltrexone, and disorbitol. And this was in CMT1A, and the goal was to lower PMP22 expression, which we know that too much PMP22 through various proposed mechanisms causes the disease. And so what I want to call your attention to is at the time that the study was being developed, the outcome that was chosen was the overall neuropathy limitation scale. So this gets back to what John was saying about how important it is to choose an outcome that's going to show us what we really need to see. But this is what was available and what was selected at the time. And so unfortunately, despite some initial promising data, the ultimate outcome of the study was negative. But I think that, I can't overemphasize how important it is that we pause and think about what this trial taught us in order to inform next steps. I'm actually going to read this. This is from the CMTA website where they summarized these findings. And again, CMTA, the website, hugely, hugely resourceful for patients to look at this. So the company said in a news release that analyzing the results was challenging because of the slow progression of CMT1A and the subjective nature of some of the clinical assessments. Those assessments can be influenced by placebo and training effects, making it difficult to determine the impact of treatment. I just want to pause there for a second, right? So what they're saying is the study's negative, the drug doesn't seem to have worked, and there's this enormous challenge, right? That we just don't have the right instrument necessarily to measure disease. So then this raises this hugely important question of, are we ready for clinical trials? If we have a drug that we really think is going to work, a gene therapy, a PMP22 lowering therapy, are we ready to test it in humans, right? And this is something that was tackled and is continually being tackled by the Inherited Neuropathies Consortium, which is really a Herculean effort to answer this question, and where the field, I think, has in a very expedited way, moved in response to these challenges with the help of the CMTA. So what is this? This is the development of new measures, right? We just heard that one wasn't optimized towards CMT. So what are they? So the goal here is to have a different set of outcomes that measure disease progression. The CMT examination score is the top one. That's you tell us how your symptoms feel. We look at your exam. We put the two together, okay? Pediatric scale, that's for kids, right? Functional outcome measure in adults is a little different. That's let's see how people are able to put pigs in little holes, how quickly they can stand up from sitting. Let's have some objectivity rather than just the clinician sort of saying, I think that you're a little stronger than the last time, right? Like, so maybe things we can time, measure, objectify. The health index is a patient-reported measure of disability, which are increasingly, these patient-reported measures are hugely important for the FDA and for us as, clinician researchers. And then there's some added ones at the bottom that I'll skip in the interest of time. So let me show you a couple examples of how the Inherited Neuropathy Consortium succeeded in taking these developed instruments, optimizing them, and then using them in studying the natural history. So this is a study of CMT1A that I actually was involved in. And I want you to look at the first bullet point. This is CMT1A. There were over 1,000 patients in this study. Okay, we're talking about a relatively rare disease. You know, you guys have gathered from the region, right? We clearly don't have 1,000 people here. You know, this was a worldwide effort from many different centers, and that's why we had the power to really look at these instruments the right way. And what we found is that there was gradual progression on our CMT examination score, so that was positive, but it was very modest progression. And this is over, 5 years, 6 years. So if we're looking at a 2-year trial, are we really going to see enough of what we need to see in progression versus those on treatment not having that progression? We also learned from this study something important, which is that the most pronounced progression we saw on this instrument was in the mildly affected patients. So that is an insight that has kind of transformed how we're going to plan for a clinical trial. Who's going to be in the initial clinical trial? Well, we want it to be the people who are changing the most at that moment so that if we halt or slow the disease, it's going to be more obvious, right? So that informs how we design the study. And here's another example. So another demyelinating neuropathy, MPZ, also known as CMT1B. And we had less than 1,000. It's not as common as CMT1A, but still a really robust number of patients. We saw the gradual progression that you see in that graph. And what insight was from this study that was really important is if you look at those two lines, the blue one is the one that's sort of changing more. And so what that is those were our axonal patients with MPZ neuropathy, because MPZ is a strange gene. It can cause a demyelinating neuropathy, the young onset, and axonal neuropathy in adults. And we don't really fully know why. But what we found when we parsed this is actually our demyelinating patients weren't changing in adulthood. They had probably already progressed earlier in life and they were sort of steady now. And so if we're going to use this instrument in a clinical trial, we have to know that we're not anticipating change in that population, right? So that's, again, something we would never know without the participation of those patients. Okay, so then I'm going to just shift to something else, which is we're not done with developing these instruments. We need additional clinical outcomes for specific types. And this brings up HMPP, hereditary neuropathy with liability to pressure palsies. I know there are people in this room because they've spoken to me with this condition. And this one's different. So this is a deletion in PMP22. There's not enough PMP22. And it presents with a very distinct clinical presentation where people have relapsing remitting symptoms. They have a foot drop, a wrist drop, a patch of numbness, and then it goes away or it gets better. That is not CMT1A, right? That's very different. And so we can't actually apply our established instruments to this disease. So what do we need to do? We need to make a new outcome measure. And this is one of these moments of honest, academic integrity of you need to be really clear about what you're going to be using to study this. And there actually is a treatment target for HMPP, which is PAK2. This is the work of Jun Lee largely. And I recently spoke to him and he said, my barrier is that we don't have a good outcome. And actually, with the help of the London group, I've developed an outcome that we're going to pilot. And this is sort of, these are the next steps. So I just want to make sure we know we're not done and not complacent. Okay, switching quickly to biomarkers. John already nicely kind of outlined this. But the idea here is we don't want to only rely on the report of a patient and the report of the doctor. We want something that's fully objective, right? That really can't be influenced by recall, by placebo effects, that's just separate from the conversation that we're having. And that brings us to MRI. So Mary Riley and Jasper Morrow in the UK have led this effort. And what they capitalized on is that when there's nerve degeneration, there is fatty replacement in muscle. The muscle is not happy because it's not innervated by healthy nerve, and it accumulates fat. And what you could do with MRIs, you can actually see how much fat there is in a muscle and very carefully quantify that. And so if you look at this graph, people with CMT1A in their calf muscle, what you're seeing there, the blue one, is that they have more fat accumulation in their calf. And over the course of a year, that muscle accumulation of fat changes faster than any other outcome we have. Faster than you can tell us your symptoms are changing, faster than we can see them change at the bedside. And so ideally, if we can show that this correlates to clinical importance, this could be our outcome potentially for a clinical trial. So then how do we put this all together? If we were doing a trial tomorrow, what would we use? Well, we'd probably want to use everything we have, right? And so that would be the clinical outcome measures that I've mentioned through the Inherited Neuropathy Consortium's efforts, plus the objective biomarkers such as MRI or something you can measure in the blood. And you want to put it all together, okay, including the patient-reported outcomes as well. And so to figure out how that's going to happen, David Herman and others have established the ACT-CMT trial, Accelerate Clinical Trials in CMT, which is meant over 3 years to examine all of these outcomes together and ask the question of how do they correlate with each other? How do they interact? What is the minimally important change we'd be looking for in any individual outcome? And also, how many patients would we need given the limitations of what these outcomes can show us? So this trial is sort of a paradigm for how we're going to prepare for a clinical trial and be as informed as possible. And I think is a hugely important study from which data is now starting to be generated. So lastly, how many patients do we need? Okay, so I'm going to give you some examples. For a CMT1A study, if we were going to halt progression 50%, and I'm not saying 100%, as you can see on the right, there's 100%, but that would mean the drug works so well that the disease is just done. Okay, so we're not going to be that ambitious. We're going to say that in the initial phase of gene therapy development, we may not get it 100% right. Okay, so we're going to say 50%. So if we were relying on the CMT examination score that I showed you early in the talk, or the functional measure, where we're measuring people's gait and things like that, we are looking at hundreds of people in each arm of a trial, and that is a big ask. But what if we add the MRI? And so if we look at the calf MRI in a particular subset of patients who we know are showing the most change, those numbers dramatically drop. Okay, so these are the questions that as a CMT research community we're faced with tackling now before any trial starts, right? Like a few years ahead of a trial to make sure that we're being realistic. All right. So what are the next steps? So as I see it, we're at a place of incredible excitement, but some of these molecules and therapies have to be translated to bigger models because we're not mice, and eventually to people. So that's really the goal, right? To bring this to patients like you. What do we need to be ready? What is trial readiness look like? One is to look at these clinical assessments that I've shown you in the talk over time, which is being addressed by the ACT-CMT trial. But the other is to develop new outcomes, such as for HMPP, but also for other atypical complex phenotypes. We heard yesterday from patients who said, I have gastrointestinal involvement, I have ocular involvement, right? Like there's going to be idiosyncratic needs for each subtype. And finally, we need to keep discovering genes. There are people in this room who have a demyelinating CMT and they don't know what they have. They don't have CMT1A, right? And there's sort of a tension around, well, the CMT1A folks get, to maybe have a treatment, and what about everybody else? And I think the promise of continued gene discovery is enormous, but we need your help to continue to generate that data. So I want to thank you, and I also want to just emphasize again that there is no clinical research without patients. Okay, there's lab research without patients, but it doesn't come to patients. And so without the help of people like you being here and learning about this, we'd be in the dark ages, right? And instead, we're on the brink, I think, of something incredibly, incredibly important. Thank you.
Good morning, everybody. Can you hear me okay? Great. I'm Rachel Salzman. I'm the CEO of Arminis Bio. And I'm going to try and not spend too much time today because I think that Dr. Svaren and Dr. Friedman already did an excellent job kind of setting this up. Arminis Bio is developing a therapy for CMT1A. We're not the only company. We're not the only technology, but I think this is just a good example to help kind of level set where we are right now. I'd like to really thank the organizers for having me here today, and I'd really like to thank everyone for coming here and taking their valuable time to spend it. The drug that we're developing, the name for it right now is TVR-110, and it's a vectorized RNAi, RNA interference. which ultimately is a controlled precision medicine approach for CMT1A. Want to just talk to you a little bit about the company, and then a little bit about the drug, which is not yet in humans, and then a little bit about the underlying technology, because as John mentioned, there's a read-through to other programs that are going to be using similar technologies. So what we're doing is a vectorized RNAi technology, and that means that we're going to be interfering with the RNA, which ultimately will bring down the PMP22 levels. This is for CMT1A, and we're doing that via a specific engineered microRNA. Your body, all of our bodies, each cell has probably 1,800, 2,000 microRNAs. We all know every cell has the whole genome, but all those genes aren't being expressed. They're being regulated, and microRNAs play a very important role. So what we do is we have engineered microRNAs that are very specific to whatever gene we're targeting. So they're very, very clean. Our lead program actually is in CMT1A, although we are also looking at targeting other genes for other neuromuscular diseases. And then what's really important when you're developing a drug in rare disease, I neglected to tell you, I am trained as a veterinarian, but I've spent 20 years in rare disease and neurology and neuromuscular drug development because my own family is impacted by a different neurological disease. So I've been living in this world for a very long time. So we have both the expertise within the company of knowing how to get a drug developed, and then we're partnered with some of the best scientists who understand the underlying research that needs to take place, namely the Scott Harbour Lab at Nationwide Children's and Cleopas Cleopa at Cyprus Institute of Neurology and Genetics, and I'm sure you're familiar with those groups. So again, a little bit more about our technology. It's vectorized RNAi. And the RNAi is done through that engineered microRNA. And I think the important thing to remember is it's like teeny, teeny, tiny microRNA that is engineered only to make PMP22 go down. And it's utilizing the innate biology and the machinery within the cell because there's all those other thousands of microRNAs that are regulating all these other genes at any given time that are keeping us going in many other ways. The way that we're getting that microRNA, or what we call a payload, into the gene is utilizing viral vector. In this case, the name of that viral vector is AAV. That's adeno-associated virus. Not all gene therapies that you read about use AAV, but a lot of them do. Most of them do, I'm going to say at this point, at least for non-cancer indications. And that's because viruses are really, really good at infecting cells, right? Viruses have been around for millions of years, and they know how to get everywhere. Obviously, this virus that we're using, AAV, it's gutted. There's nothing pathological about it. We're just reconfigured it with the payload that's going to silence that PMP22. I don't have a pointer, but that's like a cartoon of what our drug looks like. The top, that's like representing a virus, and then inside is the payload that fits in there. So at the end of the day, this is a very precision medicine product that's targeted and it's controlled delivery. Once you deliver that microRNA to the cell, it stays there for many, many, many years. We don't know that it's necessarily your entire lifetime because, we're not going to run a 40-year clinical trial, but based on animal data and other human data, we're talking about many, many years. And again, because the payload is not a gene, it's just this tiny microRNA, the payload itself is not immunogenic, which is really important in terms of safety features with, Sue talked about the importance of safety in drugs earlier this morning. The punchline here is if you don't have CMT1A, you're at the unaffected. You have protein at the very bottom there. That's sort of a normal amount of PMP22. If you do have CMT1A, you have an extra copy of the PMP22 gene. So you have too much PMP22 protein, which is, Dr. Svaren explained, is toxic to the myelin. So where our drug comes in, where it says TVR110, it interferes from where the DNA ultimately gets transcribed and translated into the protein, and it prevents it at that post-transcriptional level so that you're back down to that more normal, healthy level of PMP22. Also, I'm around. Feel free to ask me questions, and also all the other scientists can speak to this technology as well. I'm not going to go through all these data, but again, has already been explained multiple times. You have to first convince yourself, let alone the community and external experts, that this drug that I just described to you is indeed able to do what we think it can do. And this is sort of a little outline of the chapters that we've gone through over the last many, many years that have convinced us that, yes, this is the right drug that is successfully going to be able to do this, at least based on what we know so far. So in other words, we've been able to show that in the mouse model of CMT1A, we're able to bring the PMP22 down. We've also been able to show that some of the biomarkers that have been mentioned have also been normalized. When you look at the way that the nerve looks and the health of the myelin, that also gets healthy. It stays healthy. That's where the durability comes in. And then when you develop a drug, you need to have a dose response. If you give different amounts, you get different responses. And then back to delivery. We've also done a pretty robust experiment in large animals, non-human primates, monkeys, where we're able to show that the drug actually does get to those peripheral nerves, in the extremities, whether they're proximal or distal extremities, so that the drug can get there. And I'm just going to show you a little bit of data. So the point here is that we done a lot of experiments, a lot of data, and this is some of the key takeaways from it. We're able to show that the nerves that are impacted in people who are living with CMT1A, which are the peripheral nerves like the median, the femoral, the ulnar, the sciatic, that when we dose our drug, and there's that little cartoon on the left with the needle going in very bottom here. So the drug is being delivered one time to the cerebral spinal fluid, sort of at the base of the spine. It's called intrathecal route of administration. And what happens is, and that's what this data show is, even though that drug is only injected one time to the base of that spine, and in this case it was done not just in mice but also in monkeys, the drug does get all the way to the peripheral nerves, and it does bring down the levels of the PMP22. The largest nerve in the body, not just in humans, but in most animals, is the sciatic. And we're able to show that we're getting reduction of the PMP22, both in the closer places of the sciatic and the more distal places of the sciatic. Like the drug really does get there. Pay attention to the thermometer there, because I think that actually both Dr. Friedman and Dr. Svaren talked about HMPP. You can have too much PMP22, which is bad, and you can have too little PMP22, which is bad. So we call that a little bit of a Goldilocks thing. And when we look at all of our data of how much we were able to bring down that PMP22 across multiple tissues and multiple animals, we're in that therapeutic range where you see where that red bar is hovering around that 100%. I think here we're saying between 66 and 93%. And then, of course, and this is a question that you sort of are asking yourself even before I'm showing you the PMP22 levels, were the animals okay? And yes, the animals were okay. We did very, very extensive safety studies. And one of the adverse events that can happen with some gene therapies, it can be a little inflammatory to the liver. And once any tissue is inflamed, cells break down and enzymes are released. So this is where we're looking at liver enzymes in the bottom there. And when you're looking at control, which is the gray, and high dose and low dose animals, so in other words, control, they didn't even get the drug, right? All of the liver enzymes were fine, and the histopath tissues are just showing there really was no toxicity. So far, again, this is all animal data, but the safety is quite robust. And then the other point here is we want to make sure that we didn't overcorrect, because, of course, we don't want to turn CMT1A to HMPP. And in no cases in the mice or in the monkeys did it go down to 100%. The pictures on the left, those are all pictures of myelin, and they're actually all pictures of healthy myelin. And then you're looking at the function of the nerves. So at no point did any of these animals exhibit kind of what I would call maybe an overcorrection. So we're able to bring down that PMP22 to where it needed to be without overdoing it. And then just a step back about this underlying technology, because you hear a lot about AAV and gene therapy. And again, I sort of want to emphasize, I think one day we might have other ways to get payloads into, it's my last slide. One day we might have other ways to get payloads into cells, but right now viruses are the best of infecting them. AAV has been delivered to thousands of patients by multiple routes of administration, including intrathecal. We're understanding more about the safety and tolerability. Intrathecal route of administration that I described to you all has not really been associated with any adverse events. When you're hearing about the more adverse events, it's when people are getting it IV. And there's also ways to bring these therapies forward where you don't need hundreds of patients. The FDA is very supportive of these one-time precision therapies so that you can get accelerated approvals and get to market quicker than some other types of technologies. Not going to go through this, but this way it'll be there for an enduring material that you can look at online. The benefits of an AAV drug, it's given once, and other drugs, you need to repeat them over time, but then the benefit is you're only having to be in the treatment center one time and you're monitored, and then the burden on the patient is very little after that. So I think with that, I'm just going to let you know where we are right now. We finished our preclinical work. We do have orphan drug designation and rare pediatric designation, commitment both to pediatric population of CMT1A patients, which I think is important. Completed our pre-IND with FDA, are doing our manufacturing and getting ready for IND and to dose patients. So sorry it took a little time, but I'm really looking forward to the discussion. Thank you for your attention.
Have this mic. Thank you, Rachel. We've now got 20 minutes for Q&A. So we've got a great panel up here. What Rachel didn't mention, and I think, is that CMTA has a general strategy to invest in sponsored research within academic institutions and universities. But last year in 2023, the CMTA made its first company investment, and that was in Armatus Bio. So the CMTA, it's public, it's in the public space, so I can talk about it. Made a $200,000 investment in Armatus Bio as the so far most advanced genetic therapy in CMT. So we're all crossing our fingers, and it's really been a pleasure working with Armatus, who've been keeping us informed every step of the way with their data and how their studies are going. And we're all cheering them on and hoping that everything keeps working positively for them. So I'm going to open to the floor. You've got me as your runner. So if you pop your hand in the air, if you've got a question, I'll come over to you with the microphone. Sir, I saw you first.
So first, thank you all for taking the time this weekend to come out. These have been really great to hear from all of you. So this is a question for Dr. Salzman. Have you publicly guided on when you might do an FDA or when you first expect to dose a patient in the phase 1 trial?
No, we haven't yet. We still have to have our IND, and you still have to know what's going to happen during that conversation. So not yet, no.
Thank you, Rachel. I'm going to go here. Chris is bringing the mic from behind. Thanks, Chris.
Hi. What is your opinion of what will happen to the muscles served by the nerves that possibly will, I don't know if I want to say increase myelation, but at least stop the demyelination?
Are you asking about the specific drug we just heard about or just in general?
There's specific one, I think.
Okay, so based on our mice data, which are based on mice that have CMT1A, we're actually able to show in the case of the mice where their nerves actually not just look better, but act better. And then the way that the mice behave, which is kind of maybe like people with CMT1A, they actually function better and their nerve conduction velocity improves as well. So we're taking the very optimistic approach that some level of that will manifest in humans. So in other words, there will be at least decrease of progression, halting, and maybe some improvement, because in the mice, they actually got better.
Yeah, I can just add to that, like from a clinical standpoint, I think we can't know what's going to happen in people. I think the anticipation would be that if you have a very severely atrophied, affected muscle in someone who's had chronic weakness for years, you're not going to expect reversal of that muscle function, okay? But anything that's viable where there might be a reduction in function, but the muscle is still undergoing degeneration and fatty replacement, like I mentioned in the talk, you might hope to slow, halt, potentially that, and then the muscle strength would then potentially improve even a little bit, right? Depending on the timing of when you treat, which brings up another hugely important issue of like, when are we treating in the disease course, right? So Are we trying to, treat people early on? Do we want to look and see what happens later? Sometimes we're surprised that things actually look better later on than we expect. But ideally, all of us want to dose people basically in utero, right, before they develop symptoms.
Thank you. We're going to go to Zoom. We have a question from one of our hybrid participants.
How can Canadians become candidates in trials? Is there an age criteria?
I think in terms of defining which patients will enter clinical trials, this is going to be a dialogue between the clinical community and the company itself. It depends a little bit on the therapy, and there may be different staging criteria as well. So I don't think there's a hard and fast rules at this point, and it's probably going to be different for every type of clinical trial that we're going to have.
Any questions? Thanks, Grace.
My family has 2 types of CMT. We have 1X and 1F. I ended up with 1F and MS. Is there any correlation with MS and CMT?
Wow, that's a hard one. So I can tell you that I have a few patients who have MS and CMT, and I've been doing, periodic literature reviews. I think white matter changes, spinal cord involvement, lesions can be associated with certain forms of CMT. The full-blown MS syndrome, in its kind of classic sense, I am finding to be less compelling with a genetic change in a CMT causing gene. The overlap of upper motor neuron and lower motor neuron involvement exists in different genetic neurologic disorders. But I think for your specific question, it's a matter of learning more about your MS and, what exactly it manifests as, what's meant by multiple sclerosis. But what I will say with confidence is I have a few patients where we're faced with this exact conundrum, and feel free to find me in the break and we can talk about their mutation and yours if you're interested.
Thank you. We're going to go to this side of the room. Thomas?
I wanted to ask Rachel and John, given that AAV is dosage dependent, have you established clear cutoffs for PMP22 expression in regard to symptom presentation, or is it more of a continuous scale?
I want to make sure I understand the question. Just maybe either reword it or ask it a little differently. Make sure I want to understand you.
Yeah, you presented that thermometer a little bit earlier, and I was curious if we had established clear cutoffs for like HNPP symptoms and CMT1A symptoms, like in regard to PMP22 expression.
Okay, I'll make a comment, and then I think John will probably clarify it a little bit. So I think while it's true that people with CMT1A have an extra copy of that PMP22 gene, were you to measure their levels of PMP22 protein across every single nerve of their body, you wouldn't get the exact same number. So what we're trying to do and model based on what we've seen in our animal data is make sure that we're in that range, because that's just how biology is. It's just a little bit heterogeneous, that we're in that Goldilocks zone and we feel that we're in that zone enough. And then maybe, John, you can comment on level of PMP22 and pathology or phenotype.
Yeah, that's a really good question. In human clinical data, we only have data for 150%, which is CMT1A. We only have data at 50%, which is HNPP. And, then other people have 100%. So I think the scientific consensus is that probably lowering PMP22, like between 15 to 40% is going to be enough. And I think one of the things that I've been actively working on is really developing assays that we can use to establish that level in patient samples. And we're hoping that the assays that we develop are going to help us eliminate an overshoot, that where we're lowering it too much as well. So there are ways to assess that, and that'll be employed in clinical trials to make sure that we really have the right dose. But I would say that we don't have an exact threshold of where we need to be. But I think from the data that was shown, I think we are in the right range based on the non-human primate studies. And when I saw that data, I said, woohoo!
Thanks, John. We're going to go one more question here, then I'm going to go back over to that side of the room.
Okay, do you think that is there a role for stem cells treatment in CMT? And if yes, what to expect the best results of kind?
I'm sorry, I didn't quite hear the question. Yeah.
Is there a role for stem cells in treatment for CMT?
Is there a hope for stem cells in the treatment of CMT?
Yes, and if yes, what kind of we expect best results?
Yeah, so the CMTA has invested in some stem cell projects based on iPSC technology, and we developed cell lines for many types of CMT. Our initial intention was actually to develop those as models where we could do testing of therapies or testing of different mechanisms. Along the way, there have been some companies that have been injecting different kinds of stem cells into nerve or other places as well. There is a Korean company right now that's actively injecting a modified form of a stem cell as well, and they're actually conducting some clinical trials in Korea. So this has not been sponsored by the CMTA. And most of our projects have been really oriented around trying to use stem cells as a mechanism of drug testing so far. But there are efforts of other companies that are, trying to use stem cells. Now, most of the time when you inject stem cells, you don't expect them necessarily to persist in the body for a long term. And so this would probably be something, if it is successful, you'd still have to get repetitive injections over time. But it remains to be seen how that'll be effective or not.
Thanks, John. And we are talking to those companies. They do come and present to our STAR advisory board and let us know their data so we can keep across what they're doing and appraise it as a method of treatment. Over that side, Chris, thank you.
Hi. So I was wondering about the length of time for clinical trials. And so how you select how long it is for the patients participating, but then the broader kind of timeline for what that looks like. I know it's a bigger question with approvals. But I'm curious if there's any general trends.
Thanks for that question. It's a really important question. So in general, clinical trials are roughly 2 years. Okay. Now, the ACT-CMT trial, one of its main missions is to determine sort of what the treatment effect is anticipated to be and how it's going to be powered. And then how many patients you want in there impacts how long potentially you're going to treat. My bias, and I know there are people in the room who are going to maybe disagree with me, but if we can afford it, okay, and if companies are able to sustain this, and I know how challenging that is, I think our initial trials should be as long as possible. So I think 2 years is really, the ideal. There, I know, are so many challenges around that. And in Europe, I was just talking to a company where, the amount of preclinical data they have to generate to justify a 2-year trial already takes like a year, which is a huge ask because they're investing financially into this drug and into this timing. Also, which outcomes you're going to use might impact what a company can manage. Like if you're introducing MRI, their costs can also go up. But the reason I say that I would push hard for a little bit longer on the front end is because it's also our opportunity to learn about how these outcomes perform and what things look like. And we've learned some lessons from other areas of neurology, like the ALS genetic therapy trials, where they were banking on a certain degree of change in a short period of time. ALS is a rapidly progressive disease, and it didn't quite cut it. And so in a way, it's like, going a little bit longer in the front end might save us from unnecessary like repeat studies or anything. Now, I say that as an academic clinician, and I completely appreciate the challenges of that. But the other thing I'll say is we have an ongoing, almost completed 2-year clinical trial in SWORD. And I think every one of these studies informs, we may learn, okay, we can get away with a little bit less if we have certain outcomes that are really robust. So we're sort of in a learning curve right now.
And we do run in the risk where if a trial it goes for too short amount of time, then they don't see the effect in that amount of time, and it then ends up being a waste of a trial. But also, I think it differs depending on the mechanism of action of the drug. So some of the genetic therapies, we want to watch them for a couple of years. But we were talking with NMD Pharma about their treatment period. If a drug can start to work within an hour of taking it, then you might not need to wait for the full 2 years to monitor a patient's progression. So it will depend on the drug. So there's a lot of variables that determine how long a trial is. Everybody put their hands up again so I can see where the people who are waiting are. Gentleman here, thanks, Kim. And I can see you at the back, so we'll come to you next.
Thanks. Just wondering if there are any other HNPP in this group.
If you have
raise your
HNPP
hands?
and you're happy to, please raise your hand.
Whoa, nobody?
We just got one HNPP person. You're very special, sir.
Wow. Okay, I guess we're not going to meet after 10:30 here.
Yeah. At the back behind you, Chris, please.
Just to say, in my clinic, you would not be alone. I have plenty of patients with HMPP, and I think it is a difficult thing because, they have CMT, but it is different. And they come to some of these support groups, and their story is really different. And so I actually am a huge proponent of getting people with HMPP together in sort of like, a support group, because that one's tricky because there's this unanticipated symptomatology where like something can happen out of the blue, which I think adds a whole layer of challenge, for patients. And so it's good to bring you guys together, but you're not alone by any means.
So I'm going to go to where Chris is, and then I'm going to come here, and that might need to be the last two because we just got 4 minutes left. Let's see how quick your questions and answers are.
I had noticed in one of your earlier slides that there was a gene therapy going on for CMT1X. I think the name associated with it was Sarepta. I was wondering if you could give a brief update. I was also wondering if 1X has enough cases that we can develop enough traction to develop other therapies, or is it a small portion of what you're focused on?
I'm just going to interrupt you, John. I'm really sorry, but Sarepta are in the room, and she's just given me a nod. So I'm just going to take the microphone over, outing you.
Hi, thank you so much for your question. So yes, I did see it up on the slide before. I think it's really exciting. So I'm Nicole Lamarca. I'm with Sarepta Therapeutics. I'm here with patient advocacy. So we're just really right now working in animal models. So we're really excited. Our program is progressing. So that's fantastic. And we're really here to learn more about the community, learn from you, learn about outcome measures and endpoints, and really get to understand your needs as patients. So thank you so much for having us. And that's kind of our update right now.
Thank you, Sarepta.
Let me just add that in addition to those efforts, we've developed animal models for testing for companies to use within our network, and those can be made available soon. I was involved in a project in trying a new type of metabolic therapy, which unfortunately didn't work as well as we had hoped. Right now, we're assembling sort of a call for proposals for new ideas for CMT1X as well. One of the nice aspects of the mouse model that we have is it has really sort of prominent axon degeneration, which is a common element among types of CMT. So we're really hoping to gather new ideas and see if we can treat that as well. Feel free to talk to me or email me if there's any other questions that you have about that.
Kim, could we come to my friend with 1B who's waving at you?
Just one very quick thing to add to that, because you asked the question I think we didn't quite address. It's not an relatively uncommon form of CMT. We have a lot of patients with CMTX. There's a paper from Chris Rickard in the UK that you might be interested in looking at the natural history of the patients within the Inherited Neuropathy Consortium. It's a gorgeous paper, and they did a wonderful job. And it'll let you appreciate that, there's a lot of patients. It's not right.
Our data shows as well it's in the top 5 most common types of CMT. So 1X is right up there in our vision and sights.
So you were talking about 2-year clinical trial. This is my first summit, and it's been very motivating. What would the cost of something like that would be?
So it, of course, depends on what kind of drug and what kind of trial and how many patients. So the first answer to your question is a lot of money. And by a lot of money, I mean millions and millions and millions and millions of dollars. But there's also just quick education that Sue gave us. It's not one clinical trial. You have to do phase 1, phase 2, phase 3. So just big picture, to get a drug approved in general is over $1 billion, okay? It's not going to cost us $1 billion to get the drug because we have very efficient ways, particularly in gene therapy where we partner with FDA. So you're still talking, though, many millions of dollars. So, yeah. leave it at that.
Yeah, we can take one more. Thank you.
Thank you very much. So my first summit as well. Among the possible solutions that we're learning right now, I heard one about the development of a drug by NMD Pharma, which is they're already approved by the FDA for phase 2 clinical trial. I'm wondering if this type of medicine, it's similar, is going to come sooner than gene therapy or gene editing. Among the three of us, what is the most recommended?
I just want to make one comment and then John can speak to it. I think the other point that I really want to get across is this is not, nothing here we're talking about is mutually exclusive. So I'm not saying gene therapy instead of this, this, and this. I don't think anybody's saying that. So I think very much, even in SMA, a lot of patients are on cocktails of drugs in order to get the maximal clinical benefit. So that, I think, is a really important sort of ticket to admission, that ideally there's going to be multiple solutions that are going to be most suited for individual patients. And you can speak to it.
Yeah, so NMD Pharma is a company that really has developed an exciting drug. It's not directed at nerve, which is the source of the neuropathy, nor directly at muscle, but at the junction of those, which is the neuromuscular junction. And so a lot of us think that it does have promise not to cure CMT, but maybe to help alleviate some of the muscle weakness and symptoms. So we've been partnering with them to see if we can help deliver that drug to a clinical trial for neuropathy. And it may come as a one treatment, or perhaps it may be combined with other treatments that are in process as well. So I would say there's a lot of excitement about that, but it probably is not, we wouldn't call it a cure of CMT.
Okay, just as that microphone's been handed over, NMD Pharma are here. They're presenting now in the type 2 breakout. So if you go back and listen to the recording, whatever they say in the type 2 breakout is going to be applicable to the people in this room, because their trial is going to cover type 1 and type 2 patients. So listen back to that. Vera.
Yeah, I was just going to say, I think one really important point about these drugs that act like at the neuromuscular junction or prevent axonal degeneration, but address more ubiquitous, highly widespread nerve-related pathways that are not specific to a gene, are going to be really important. And they're not just important for CMT1A in addition to gene therapy. They're important for all the people who have undiagnosed CMT. We know you have a CMT2, but we don't know which it is. We need drugs for those people that will alleviate disease while we're discovering the genes and understanding the mechanisms. So I really, as we're all so excited about gene therapy and the potential for that, we have to be really investing still in those other mechanisms.
Yeah, we have to be so realistic. Sue put a slide up. There's over 160 identified subtypes of CMT. It's very unlikely there'll be 160 different gene therapies. So we're looking across the piece as to what combinational therapies, what might work. 40 to 50% of people with CMT don't have a genetic diagnosis. There needs to be a treatment for them. So the CMTA, that's why we have that broad portfolio approach to treatment development, because we don't just want to treat one type of CMT. We want every patient with CMT to have a treatment option available to them. With that, because of time, I'm going to say thank you ever so much to John, Vera, and Rachel for joining us today.